So is there any questions from last time before we get going? Okay. So metabolism is essentially just chemistry going on in a living system. And what we do with metabolism is it's transforming matter and energy. So chemical reactions take one chemical and make it into a different chemical in there. And they're all going to be subject to the laws of thermodynamics in there. We organize things into metabolic pathways in biology. And with a metabolic pathway, it's something where it's set out, you start with a big specific molecule and you put it through a series of steps to end up with your product, which is going to be a different molecule. And each step is going to be catalyzed by a specific enzyme. For animal cells, they're pretty much all going to go off of one main pathway for our energy metabolism. And then other things will join into that. But you look at the metabolism of a carbohydrate in the form of glucose, and if it's any other carbohydrate, it has other pathways it will go through to form glucose or join into that cycle in there. So while it's going to seem like we're only talking about glucose and nothing else seems going to sound like it's important, it's because everything else just goes through a few steps to join this same pathway as glucose. So that is one area where the body simplified things for us in that sense. So the catabolic pathways are going to be the ones that break things down, and they're going to release energy. So when you eat your food, it's going to go through catabolism. It's going to be broken down, and then you release the energy from your food. Anabolic pathways are going to consume energy, and they're going to be used to build things. So if you're going to build muscle in your body, you're going to have to use energy to do that. And bioenergetics is just the study of how energy flows through living organisms. So how energy flows through humans and animals, how we take energy from plants, and energy is recycled throughout the system. One of the things that we see is that it is all recycled through. You take your products as a human, we consume plant matter and other animals, then we have waste products that feed something else. That's going to feed plants. Plants are going to feed off of the waste products and the sunlight, and then we turn around and eat the plants. So it does form this continuous cycle in there. Energy is just the capacity to cause change, and it comes in different forms. A couple of these we've already talked about. Kinetic energy is your energy of motion. Heat or thermal energy is the kinetic energy that's associated with the movement of the atoms or the molecules in there. So an atom is going to have some kinetic energy because it's got electrons that are moving around in it. Light is also a form of energy. And then potential energy is due to location and structure. It's work that could be released out of something in there is why it's called potential energy. So with chemical energy, the potential energy is available for release in a chemical reaction in there. So if you consume glucose, it has some potential energy that's going to be released as you go through all of the steps of breaking it down. So thermodynamics is the study of energy transformation. And when we look at this, the system is going to be the matter that's under study, and everything outside the system is the surroundings. So in an isolated system, you're unable to change the matter or the energy with the surroundings. It doesn't, when it's isolated, it's completely isolated from its surroundings, and its surroundings cannot contribute or take away from the reactions that are going on. With an open system, energy and matter can transfer between the system and the surroundings. So do you think we participate, our body is going to be involved with open system reactions, or is it an isolated system? We're going to, all of us as a whole, it's actually going to be an open system because it's going to interact with the oxygen in your atmosphere. So you would feel a difference in it if you went up to Mount Rainier and attempted to do aerobic metabolism because of there being less involved in there. Isolated systems are actually kind of hard to create in there where things don't interact with their surroundings. You pretty much have to create those in a lab. So the first law of thermodynamics states that energy cannot be created nor destroyed, but it can be transferred and transformed. It's also called conservation of energy in there, which basically means you can move energy to different places, but you're not going to have more net energy or less net energy on the planet in there. So it also lets you know that you can't make something out of nothing in there, and if you take something into your body, it's going to be accounted for. It can't just disappear into nothing. The second law of thermodynamics states during the transfer or transformation, some energy becomes unusable, and it's usually going to be in the form of heat, and it ends up making the universe more disordered. So this idea where you start saying the universe becoming more disordered, try not to overthink that and let that idea get to be confusing to you. Basically, when you break something down, if you cook it, you're going to release some of the energy as heat. You can't get that back in there, but it is not gone. Instead, what it does is it creates more disorder, more movement in the universe, and that's what we mean by disordered, is it goes off into the universe, we can no longer harness it, the energy's not gone, it's just out there. So entropy is that measure of disorder or randomness that from every energy transfer or transformation is going to increase the entropy of the universe. So it would have to be 100% efficient in order not to increase entropy in the universe. And there really is no set of reactions that are going to be 100% efficient where no heat or energy is lost to the outside world in there. So increased entropy is evident in the physical disintegration of systems of organized structure. And then spontaneous processes occur without the input of energy. And for a process to occur spontaneously, it must increase the entropy of the universe. So in order for something to be spontaneous, it has to go into a more stable state, and it's going to release some of that instability into the universe to increase the entropy in there. You're not going to naturally progress towards being in a more unstable or an uncomfortable position. As your day progresses, you want to move into a more and more comfortable position. Any chemical substance is going to want to move into the lower energy state naturally, and the difference between the two has to be accounted for, and it goes into entropy in there. So biological order and disorder. Living systems increase entropy of their surroundings. It takes a more ordered form of surroundings and replaces them with a less ordered form of it. So we can actually measure this energy in there, or this change in energy, as the free energy, and the amount of free energy change is going to determine if a reaction occurs spontaneously, or it will tell you how much energy is needed to put into it in order to get it to occur. And so we call it the delta G, or Gibbs free energy. And it's the portion of a system that can perform work when the temperature and pressure are uniform in a system. So when we talk about these things, the theory of them, we're always assuming uniform temperature and pressure. However, when they're actually in a living system, those things are not going to be uniform. So that's how you'll get different numbers in there, is every living system varies a little bit in there. So the situation it's in will vary. So the equation that goes behind it is delta G equals the delta H minus T delta S. Delta H is the change in the enthalpy, which is going to be more of your heat. Delta S is the change in the system's entropy, and the temperature is the absolute temperature in Kelvin. This is one of those equations that we won't actually be calculating anything using this equation. It's more to give you the theory behind it as to what all the components are and determining the free energy of something. But what we're going to be more interested in is looking at, is that free energy a positive or negative number in there, because that's going to tell us, is this reaction going to occur or not. So the delta G must be negative for the process to be spontaneous in there. If you are to multiply and complete all of the reactions in there. So what we look at with something is your change in the G is your free energy of the final state minus the free energy of the initial state. That is what delta G is. So if you look at how much energy is in this system at the beginning and how much is in it at the end, that difference is your delta G, or free energy. The delta in there, when you see that little triangle symbol in front of it, it means the difference in something, if you haven't seen that notation before. So delta G is only going to be negative when the process involves a loss of free energy in there. And things change to a more stable state with a lower delta G. So as a reaction is going to proceed towards equilibrium, the free energy of the mixture of the reactants and products decreases and increases as it moves away from equilibrium. So as you get closer to equilibrium, that delta G gets smaller. As you get farther away from equilibrium, it gets bigger, because it always wants to go to equilibrium. You're working against things when you go away from equilibrium, so you would have to put in energy. When you go towards equilibrium, you're letting what naturally occurs happen, and so that's going to lower the energy in there. Does that idea make sense? Yeah, basically it always wants to come back to stable in there, and to maintain something outside of that is going to be a lot more work. So with free energy in metabolism, the exergonic reactions are going to proceed with a net release of energy. So they're going to be ones that release energy, and they've got a negative delta G. And the magnitude of delta G represents how much work it can perform. So with cellular respiration, what this C6H12O6 represents here is glucose. It's a chemical formula for glucose. When you take one glucose and you mix it with 6 oxygens, it's going to break down and give you 6 carbon dioxides and 6 waters. This over here, the carbon dioxide in water, is a more stable state. So it's going to naturally want to go there. So the delta G for that is negative 686 kilocalories per mole. If you wanted to look at it in joules, the number is negative 2870 kilojoules per mole. So for each mole of glucose, which remember that mole is 6.02 times 10 to the 23rd of those atoms in there, it comes out to be 180 grams. For each 180 grams of glucose that's broken down under standard conditions in there, pH of 7, 25 degrees Celsius, 1 molar concentration, there's 686 kilocalories available for work under standard conditions. That is not under body conditions. So the energy of the products is 686 kilocalories per moles, less than what the reactants were. So you could think of it, if you start out with these reactants, you have these products here, plus 686 kilocalories per mole of energy. So does that make sense to you? When you break it down, you get the products and the energy out of it. So with an endergonic reaction, you have to absorb free energy from the surroundings. You have to put energy into these to make this. It essentially stores the energy in the molecules by creating the bonds in there. So the delta G is going to be non-spontaneous, and energy has to be put into these. So if you think of it being like a tree or a piece of wood in here, when you're burning that piece of wood, that would be what you'd start out with over here in the reactants. You have a pile of ashes in your products and the heat that was released from that wood. That heat you would measure in those kilocalories in there. In order to make that wood with that tree, you have to have a plant take the energy from the sunlight and the carbon from the air and use that energy to glue that carbon from the air together. So with metabolism and equilibrium, metabolism is never going to be at equilibrium. There's going to be a constant flow of materials in and out of the cells. So your cells are never going to reach equilibrium because they're constantly having to spend energy to stay alive. So they're never going to reach that balance point in there. There's always going to be a need for more stuff coming in. And you're always putting stuff in there. So we try and figure out where that equilibrium is when we try and figure out how much we need to eat to maintain our body mass in there. But for most people, that's going to fluctuate a little bit on a daily basis because sometimes you're going to eat a little too much. Sometimes you might not eat quite enough to sustain everything in there. So our reactions are generally going to be pulled in one direction when the cell releases the product. So our cells aren't going to build up glucose and sugar molecules. We only break them down. So it's always going to pull the chemical reaction to go in the direction of breaking it down. It's never going to go in the other direction of building it back up. So ATP comes into play here. It's going to help in the coupling of reactions. So when you break something down and you get energy out of it, you're going to use that energy to fuel some other process. So you get up in the morning, you eat breakfast, you use the energy from that breakfast to fuel you to get started with the day in there. But you have to have something that couples that because the energy, you need to be able to store some of that energy you ate from the breakfast to sustain you for the next few hours in there. And how we do that and couple it is with ATP in there. So chemical work is going to push the endergonic reactions that are not spontaneous in there. So it's going to provide that energy in there for the chemicals. So sometimes you have reactions that you're going to do in your body that are not spontaneous. You're going to need some energy to fuel them. If you're going to build new muscle tissue, that's not spontaneous. You need to have some energy that you can use to fuel that process. And that's one of the ways you'll use ATP. Transport work. So with those sodium-potassium pumps in there are a good example where you're going to pump sodium across the membrane in there in a direction it doesn't really want to go. You're going to have to put some work into it to do that. And it's going to do that with ATP. You actually spend about a third of your ATP energy every day just fueling sodium-potassium pumps to move sodium out of your cells where it doesn't want to go. Mechanical work, that would be things like beating cilia, contracting skeletal muscle. You need ATP to do that. And then the energy coupling, using the exergonic processes to drive an endergonic one. You use the energy you get from one to fuel another in there. So ATP is adenosine triphosphate. You've got an adenosine molecule, and then you've got 3 phosphates attached to it in there. They're phosphate groups. And the phosphate groups are broken by hydrolysis, which means you're going to add water to it, and it's going to split the bonds in there. And then it's going to pull off the last phosphate group, and it becomes ADP for diphosphate instead of triphosphate in there. So when you take ATP and water and you cause them to split apart, the delta G on that is negative 7.3 kilocalories per mole. So it's a relatively small delta G in there compared to what you get from breaking down a mole of glucose. But this is a single molecule where the mole of glucose was 180 grams in there. So you would expect a big difference. Mm-hmm. The only one that you would probably, the one for glucose, you would want to know, the 686 in there is one of those things that's likely to come back. For the ATP, the negative 7.3, just know it in kilocalories per mole. Don't worry about knowing the joules conversion. Yeah, of all of the numbers, those are the only 2 right now. If you really wanted to figure it out in kilojoules per moles at some point, at that point you would be comfortable doing conversions between those units and you could figure it out from there. I wouldn't bother memorizing that. So that's going to be in laboratory conditions that splitting ATP gives you that. It's not cell conditions. Cell conditions are not standard. Cell conditions are actually more favorable for splitting the ATP. So you actually have a delta G that's negative 13 kilocalories per mole. It's 78% greater under cellular conditions. So it's much more efficient to do that in a cell than it is outside of a cell. And this is useful in the cell because the energy released is somewhat greater than what most other molecules involved are. So you can almost think of the ATP being like dollars or an energy currency in there that you're going to use to transfer energy of things. So how it's going to perform work? Cell proteins harness the energy from ATP hydrolysis to perform work. They're going to use that energy to perform something else. It will couple with an endergonic reaction, and it's usually going to involve phosphorylation. So when phosphorylation just means you're putting a phosphate on something. So you take the phosphate off of ATP and you stick it on somewhere else, and when you give them that phosphate, you're giving them the energy that goes with it. So transport and mechanical work is almost always powered by ATP in the cell. A lot of times the hydrolysis is going to lead to a change in the protein shape and its ability to bind another molecule. So you may be this shape when you're bound to ATP. When you split the ATP, you might be this shape in there. So not only have you released the energy, but it's causing a molecule to change shape. And so it can a lot of times do work with muscles. They will change shape. The myosin, when it grabs onto actin in muscles, changes shapes, and it does a ratcheting movement like that to shorten your muscles. So since we are going to be using this ATP stuff, we also need to be able to regenerate this ATP stuff. And it's going to come from your exergonic reactions that break things down in the cell. So one of the ways that we are going to help to control this in the cell is by using enzymes. Enzymes help to speed up metabolic reactions by lowering the energy barriers. You could think of them being like a fence that you have to get over. If you lower the fence, you make it easier to get over it in there. And that's what an enzyme is going to do. So enzymes are macromolecules. They're usually made from proteins that will act as a catalyst that will speed up a reaction without being consumed. So they lower that fence, but they don't get used up in the process of lowering that fence. That enzyme is there, and it will constantly be available to be reused. So if one of the ways that you lower that barrier of the fence is by using a set of stairs in front of it, only so many people can go over that set of stairs at a time. It's the same thing with the enzymes. Your enzyme is going to have a maximum capacity of how many molecules it can help at a time, and that will end up being what limits the rate of your reaction usually, as how much enzyme do you have to help these molecules get over this barrier. So your activation energy, or your free activation energy, is the amount of energy that is required to contort your reactant enough to break the bonds. So in an equation or an abbreviation, we call it EA in there, your energy of activation in there. So if you're going to break something, you may be strong enough to wiggle it and bend it, but you can't quite break it in there. What the activation energy of the enzyme is going to do is make it easier for you to break it in there. So what this graph represents is, up here is the amount of energy that's needed to break it. So if you can't put in enough energy to break it, it isn't going to break. If you can't reach that threshold, you'll not get to the other side of it in there. And what the summit of the graph represents is the free energy, what is going to make the reactants unstable enough that they're activated and the bonds can be broken. And in chemicals, a lot of times what's going to do that is heat. If you heat it up, you make it unstable enough that it will actually progress into that reaction. And that graph, even though it looks like it's kind of wavy like that, that's because I drew it with the word drawing thing. It's actually a smooth wave in the line. So if you think about if you want to make something out of wax and you try and mold that hard wax into the shape, it's too hard. The energy required is too great for you to be able to do that. But if you put some heat in there and you soften it, now you can move it and manipulate it in there. And that's what you do with heat in a chemical reaction. So how this is going to happen is the heat speeds up all of the reactions. It speeds up how fast everything is moving in there. And a lot of times when it does it, it's going to denature the protein so that it's not appropriate for that living system anymore, and it has to finish going the other way. So it's like when you cook an egg. If you've got egg whites, or not egg whites, but like scrambled egg mix, and you're cooking it, if you have it in warm water, like lukewarm water that you can put your hand in, you can have it in that temperature all day long, and it's not enough energy to denature the bonds. However, if you stick it in boiling water in there, it's going to denature it pretty quickly. It becomes cooked in there. So that's what you're looking at doing in there, is denaturing the protein in a lot of cases. The enzyme doesn't change the delta G, but it catalyzes the reaction by lowering the energy of activation barrier. So you still get the same amount of energy, it just makes it easier for you to get over that hump to access it. So one of the things we're going to do in the lab next week is we have a lab on enzymes, and we use papaya enzyme in there. It's bromelain from papaya juice, and you try it at different temperatures to see how it reacts with gelatin. So enzymes are going to be really helpful to have these chemical reactions go on, but they're also very picky. The substrate is going to be the reactant that the enzyme acts on. So it's going to be what it chooses to help in there. And when the enzyme binds to that substrate, it forms an enzyme substrate complex. And that's going to be what allows the catalytic actions of the enzymes to convert the substrate into its products. So when it binds to that, it's going to allow it to change into the products in there. Usually the enzyme name is going to end in ASE. And the names are named a lot of times by what they do. So you might see a phosphorylase that's going to phosphorylate something in there. The enzyme names are going to closely correspond, so you can see what goes together. A hydrolase hydrolates something in there. So the active site is going to be the restricted region of the enzyme that binds the substrate. It's usually going to have a pocket or a groove on the surface. And they're very specific. So if this enzyme is going to work on helping one type of molecule, that's the only one it will help. It usually does not help anything else. So if you looked at it, and this is your enzyme here, your substrate could fit in there. It only helps triangles. It does not help squares, circles, cubes, anything else. It only helps triangles in there. So we say it has an induced fit. A lot of times when the enzyme and the substrate come together, it's going to change shape slightly so that it fits snugly around it. So you could say that when this comes in, that it would have little arms that would come up and help hold it. So when it comes in, holds it in place, and then it can let it go when it's done with it. And a lot of times it's going to bring the active site into a position to enhance the ability to catalyze it in there. So while the substrate's held in that active site, the active site catalyzes the conversion to the product. So a lot of times it's going to bend it, and it's going to put it in close enough proximity that it can actually grab onto those other reactants it needs to become the product in there. It kind of acts like a booster, that it can now reach what it needs to in there. And a single enzyme can act on hundreds or thousands of substrates of molecules per second. So this is occurring very quickly. When we look at it on the board and we try and picture it, we're picturing it happening slowly in there. But it's actually lightning speed that this will occur. Most of them can be reversible, and the enzymes can catalyze forward or backwards in there. However, when we look in living systems, you usually have that equilibrium drawing the reactions to only go in one direction, and they don't need to be reversed. So enzymes lower the activation energy by bringing the reactants together in the proper orientation. It may stretch them towards a transition state form. The active site may provide microenvironments that are more conducive to the reaction than the solution itself was. It can direct participation of the active site in the chemical reaction. So it may have a brief covalent bonding between the substrate and the R group that will allow the subsequent steps to restore the active site in there. But you're always going to get the enzyme back in its original form at the end. So the rate at which these occur is going to be partly a function of the initial concentration of the substrate. So if once you start running low on the substrate, things are going to slow down. When you have a lot of that substrate, everywhere the enzyme looks, there's someone to help. So it's going as fast as it can in there. But after a while, when you've helped all of those and there isn't that many left, it slows down the process because, oh, okay, it's taken a little while. The line is just trickling in now in there. It will slow down in helping. At some point, the enzymes become saturated, so there is a maximum speed. If all of your enzymes are busy helping in there, and you throw more in there, it's going to have to wait. The enzymes can't go any faster. They're all busy helping in there. Where that becomes important is when you look at drug doses in the body and drug interactions. If you have two drugs that you put in the body that require the same enzyme to process it, you don't have more of that enzyme. What's going to happen is one of those drugs isn't going to be processed as quickly as it should be. And that's what's behind a lot of drug interactions and toxicity, is your liver only has so much of certain enzymes. It can only go so fast. So if you put more than it can handle, you're going to pay the price in the damages there. So what are some things that can affect the enzyme activity? The temperature and the pH. Being that they're a three-dimensional structure, they're going to be sensitive to their environment. An enzyme will have optimal conditions that are going to favor the most active shape. Generally, as you increase the temperature, they work faster. But you can increase the temperature too much that you just damage it in there. they also have an optimal pH in there. And when you take it outside of that pH, you will get to a certain point where it just won't function anymore. Another thing that will influence it is cofactors. Some enzymes are going to be complete and they're in their whole shape from the get-go. Others are going to require these cofactors, some other piece that needs to come into the puzzle in order for it to work. And these are going to be non-protein. They're usually an inorganic metal. Most of the minerals in your diet, what the role that the minerals play is they are cofactors for enzymes in your body. And then the coenzymes are our organic cofactors. Your vitamins will act as coenzymes or help to make the materials to make them in there. So one group of vitamins that is particularly important for your reactions in there that are used to derive energy are your B vitamins. Sometimes people will talk about, oh, I feel like I have more energy taking these B vitamins. And it's not that the vitamins are giving them more energy. What it's doing is making more of their enzymes available to make the energy in there. So of course it can't be that simple that you just have enzymes and they're active all the time. We have to have ways of inhibiting them. We always want to have this balance of things, and you need to have something that shuts things down. So the competitive inhibitors, they're going to mimic the substrates and they're going to compete for that active site. So if you've got your triangles that want to go to this enzyme, a competitive inhibitor may look like this. And what's going to determine who gets in there is the concentration. So if you have a lot more triangles than you do of the inhibitor, right now that inhibitor isn't going to do a whole lot to shut that down. But if we get rid of most of those triangles in there, now that inhibitor's got a better chance of getting in there and blocking it than the triangle does in there. So that's how a competitive inhibitor is going to work. A non-competitive inhibitor is going to bind the enzyme in some other site other than the active site in there, and when it does that, it's going to inhibit it from functioning. So we'll say that we've got a site over here that happens where that triangle can bind, and when it does that, it just closes that hole so that the triangle can't fit in. So they're not competing for the same site in there, but it does stop it from working on other things in there. So the way a lot of toxins and poisons work, and they're often irreversible, is they're going to inhibit enzymes. So the sarin nerve gas is an example of something that does that. A lot of pesticides will work that way, DDT and parathion. They're going to inhibit enzymes in the insects, and then a lot of antibiotics are going to work that way, where they're going to inhibit enzymes in the bacteria. So regulating the enzyme activity helps to control metabolism. I think we're almost done with this, and then we'll take a few minutes in there. The ghost is back. So allosteric regulation, this is going to be where the protein functions at one site, and it's affected by binding a regulatory molecule to a separate site in there. So these can either inhibit or stimulate things in there. So this is going to be the way that most enzymes are regulated. They're going to have 2 or more subunits that each have their own active site. And then you have to bind the activator to the regulatory site to stabilize the shape of it. So an activator would stabilize it in the active form, an inhibitor would stabilize it in the inactive form. So the fluctuation of concentration in the regulators influences the response of the enzymes. So for example, there's a bacteria that it's going to make an enzyme to process lactose only when lactose is present. So the lactose that it needs to process is going to help to stabilize that enzyme and trigger making the enzyme. It doesn't want to waste making the energy to make that enzyme if lactose isn't present. So it's a way that the cells become more efficient by regulating these things. So sometimes when you bind one active site, it can stimulate the catalysis of multi-subunit enzymes and affect other active sites. So you can have stimulating one have kind of a snowball effect where it will turn on and regulate several other enzymes in the process. So when that happens, we say we have cooperativity. So a substrate that causes an induced fit in one subunit can trigger that same favorable reaction in all the other subunits of the enzymes. And that's the way it can amplify the response. So when hemoglobin is an example of something that does this, when it binds one oxygen, it becomes more aggressive at seeking out additional oxygens. It's going to be more efficient at binding the additional oxygens. So you get a little bit of oxygen in there, it gets a taste for it, it primes it, and it's ready to bind even more oxygen in there. So identifying allosteric regulators, we don't know what a lot of these are. It's kind of difficult to identify these things chemically. We assume they're probably pretty widespread, but it's really hard to isolate these because if you take an enzyme out of the living system, it's not going to be in those same conditions to cause it to act like an enzyme. So it makes it a little hard to study. So feedback inhibition. This is another thing that you'll see where a metabolic pathway is switched off and it's inhibited by the end product of the enzymes that are early in the pathway. It says, we've made enough of this, don't make any more. It's kind of a way of looking and saying, system's full, stop making it. So the enzymes have specific locations within cells. Some of them are going to be compartmentalized and then they're brought into the place where you need them. And that arrangement can actually facilitate the sequence of reactions in there.